Good evening. Welcome to Insights, Newcastle University's Public Lectures programme. My name is Martin Farr. I'm co-chair of the Public Lectures Committee. It gives me great pleasure to welcome you to the second lecture of our spring programme. It doesn't feel very much like spring outside, so thank you for bearing the weather to be with us today. Could I ask you to ensure, please, that your phones are in a silent mode during the lecture, also for notifications. I'm not expecting a fire alarm this evening. Good luck, any fires this evening as well. If there is a fire alarm, please come and join us in the car park immediately afterwards. I'll draw your attention to, in our programme, which is available outside, our next lecture on Tuesday, Science Sans Frontiers by Sir Keith Burnett, President of the Institute of Physics. That lecture, like this evening's, will be recorded and available on our archive on the webpage. We have many hundreds there for you to watch at your leisure. Two people speaking this evening before our speaker. First of all, we have, you may have seen the stalls outside, we have Les Milne from the Humanist Celebrants of North East to say a few words. Les, please.
Thank you for that introduction. It's a great pleasure for me to be here in my capacity, as the chair of North East Humanists, and of course we're one of the largest humanist groups in the country. Been around for about 67 years, and during that time provided lots of opportunities for humanists to meet in Newcastle and in the wider region. And Darwin Lectures, presented through this Insights programme in association with the North East Humanists, or NEH, I'll probably refer to them later. It's an occasional series of lectures that mark the work of the father of evolutionary biology, Charles Darwin, and just how that piece of work that he did on the origin of species, published in 1859, the lecture does show how that piece of work has in itself evolved. And it's scheduled at this time of year, of course, because it celebrates the birth of Darwin, 12th of February, 1809, so this year will be his 215th birthday. A lot of candles. And of course, there are lots of quotes which are attributed to Darwin. And I think the one I think is most appropriate for today is from another of his books, that's The Descent of Man in 1871, where he writes, "Ignorance more frequently begets confidence than does knowledge. It is those who know little, not those who know much, who so positively assert that this or that problem will never be solved by science." And of course, given the scientific progress that's been made since he wrote those words, or indeed in every one of our own lifetimes, it's clear that science has provided solutions to many problems. And it's the search for evidence that's key to this, and through the work of academic institutions like this fine university and also the organisations such as NEH, that important evidence can be shared and put to good use for the benefit of society. And so I'm certainly looking forward to Jelena's lecture, both from my viewpoint as a humanist, but also having had the privilege of a career in public and environmental health. And of course, it's those challenges that have come through public health that I think many of which are going to be addressed by the pioneering work that she's done. And of course, to end, I really must take the opportunity to invite you all to sign up to the NEH monthly bulletin. It gives details of all the events that we have, and in which we hope to give an insight, if I may be able to borrow that word for the moment, give you an insight into humanism, and certainly my colleagues on the desk at the front will be pleased to see you. And so, in the spirit of sharing information for the benefit of society, I'm just going to close with a 6-word statement on humanism. Think for yourself, act for all. Thank you.
And now to introduce our speaker and to chair the questions and comments afterwards, Darwin Mann, Professor of Hepatology and Head of the Newcastle Fibrosis Research Group.
Thank you very much. So my job here is to give you a flavour of tonight's speaker, who is a very inspirational lady. So Jelena started her academic career by getting a PhD in cancer immunology at the University of Southampton. That's quite a difficult thing for anybody to do. But she did it while also single-handedly bringing up a young daughter. And that daughter herself, with Jelena's guidance, is becoming quite remarkable, currently running a gastroenterology unit at the Royal Free Hospital in London. So after finishing her PhD, Jelena then switched to look at chronic liver disease, which is a big public health problem. And what she brought to that was really something very different, and that was to ask how genes are controlled when your liver starts to become sick. And she genuinely revolutionized the field by opening it up, opening its eyes to this new world of epigenetics that she's going to tell us about today. And then when she moved to Newcastle, she continued to do that work and really produce some landmark work that has given her many plaudits and prizes. So that really should be enough, but in fact, she's done more than that, because in recent times, she has transformed herself into a successful businesswoman. So she's set up and is CEO of a company called Fibrefine, which is based here in the North East of England. And what that company is doing is looking for, or helping the pharmaceutical industry to look for drugs that will not only combat liver disease, but lots of other chronic diseases. And that company, although it's based in the North East, has a global presence because it works with almost every single pharmaceutical company you could possibly think about. And while doing that, she's been still juggling, doing her academic career, publishing papers, inspiring students, and also bringing up a second child, Thomas, who's here in the audience somewhere, who himself is becoming quite a remarkable individual, recently in his area of expertise, captaining the Northumbrian County under-18s rugby team. So, without further ado, I'm going to invite Jelena up to the podium and introduce her to give her talk, which I'm absolutely sure is going to entice you.
Thank you. Well, I hope that after that introduction, I don't do a terrible job and you are all really disappointed. So, Derek is absolutely right. I have done quite a lot of work on epigenetics, but it's all really been just kind of playing in the lab. I've had incredible freedom through support of my colleagues because we have always worked as a team. And they have supported me to be able to do really blue skies, crazy things. And I will show you some of that stuff today. But before I show you what we have done, it's really quite important for public to understand what genetics are, what epigenetics are, how does all of this fit together? Because a lot of times you will read about genome and DNA and this and that in papers, but I certainly know when I've spoken to my grandmother while she was alive, and I tried to tell her about the things that I was doing, and she'd be like, well, in my head, this just doesn't make any sense. I can't really see it. So then, in my talk tonight, what I'm going to do is, rather than use a lot of writing, I will use a lot of videos to kind of bring this molecular biology world to you in pictorial forms, because a picture speaks 1,000 words, and I hope you will agree with me when we go through the lecture. I hope that I will be able to bring a little bit of flavor of what I do, what I'm interested in, and also leave you a little bit more enlightened about the subject. So I think we can't really talk about epigenome or epigenetics without first understanding DNA. So this is what DNA looks like. It's a double helix, something that looks like a twisted ladder, okay? And this is a chemical that is present in every one of your cells, and it contains a blueprint or a genetic instruction how to develop and function a certain body, okay? And it tells each one of the cells what to make and how to make it. But interestingly, every single cell will have exactly the same DNA, okay? So then this blueprint is used to make proteins, which are building blocks of all of our cells. So maybe some of you have never even seen a cell. So I thought, right, this is what cells are. If you could see into the cell, you would see a lot of different parts, a lot of organelles. But that thing in the middle is a nucleus. That purple thing that we're going to dive into now is where your DNA is. Okay, so every cell, more or less, will have similar sort of thing. So this DNA is organized into something that's known as chromatin, and I will explain to you that in a minute. And that chromatin will sit in lots of different forms inside the cell, but when its cells come to divide, it will get very much condensed into chromosomes, which will then split into 2 different cells and create daughter cells, okay? So then, I have already alluded to you, how does identical genetic information, the same piece of DNA in every single cell, give rise to over 200 different cell types in 70 trillion cells in a body and 78 different organs? Quite clearly, when you look at that, and you know yourselves, you are not like an amoeba. You're not a blob of same cells. You have eyes and elbows and joints and muscles. So how does DNA do this? And the key is that while DNA inside your cells is the same, the way the cell interprets really that blueprint defines what the cell is. So the human DNA is probably about 6 feet tall, so a bit taller than me. And it has to get compacted into that tiny nucleus. Okay? You can't even see a cell with a naked eye, yet it holds something that's 6 feet long. So that poses 2 different problems. How do you package it? Obviously, not all the phones were switched off. I'll just wait until you answer that, whoever it is. So, the human DNA, that 6-foot-long strand, encodes somewhere between 20 and 25,000 different genes. So each one of the genes, in very simple terms, is a little piece of DNA that codes for one of the proteins that makes a building block of your cells. And different combinations of genes will then kind of give rise to different combinations of proteins in the cell, of different building blocks. And then what is expressed in any given cell type will define what that cell is. So let's put it very simply. If we imagine that this blue thing is the human genome, very simplified, and let's say that these little orange blocks are the genes, okay? If we choose to express this combination of genes together, we will get a cell that's a neuron that might be inside your brain. But if instead we express this different combination of genes, then what you get is an immune cell, okay? So that is how this same piece of DNA will give rise to different cell types. Now, this presents itself also in animal kingdom, where not only does this difference in how genome is interpreted give rise to different cell types, but it can actually cause a decision whether you are female or male. So in American crocodiles, alligators, genetically identical eggs are becoming male or female based solely on temperature at which they are incubated. So clearly, there's males born at 34 degrees, and if the egg is incubated at 30 degrees, it gives rise to females, which probably has something for us to think about with the global warming, that we might lose the entire species because they will all be males. Examples of this are also present in the insect world. So you will all know this is a maggot, okay, which then transforms into a fly. And clearly these two look totally different. So their phenotype, or the expression of kind of who they are, is very different, yet their genome is exactly the same. So same DNA here, but how it's interpreted is completely different. So that then brings us on to the thought that actually genetic identity is not only present at the cellular level, but it's actually present at the global phenotypic level. And we know that just by looking at ourselves. When I was a child, I didn't look like this, okay? And when I was a young woman, I was probably about 4 stone lighter and much more attractive. So we all know that aging changes how we are. We change over time. So then, if genetic identity isn't the same as phenotypic identity, both at cellular and organism level, all of these combined are epigenetic phenomena. And what epi means, it comes from a Greek word meaning or in addition to or above the genome, okay? So this is your genome, but the epigenome is something that works on top of the genome, that actually instructs how that genome is interpreted. So this is what perhaps a very simplified cartoon of what a simple kind of building block of an epigenome is. We'll go through this slowly, and then I have a video for you to watch, which will bring it to life, hopefully a little bit more. So what you see in this yellow twisted ladder, this is the DNA, okay? And I have already alluded to you that inside your cells, DNA is not naked. It's actually combined with these epigenetic marks. So the marks could be addition of certain chemical groups. This is a methyl group, let's pretend. You will see an actual methyl group in the video. But methyl groups, different chemical groups, can be added to the DNA itself. And that will carry a certain signature. Usually, if you have DNA methylation deposited on a gene, that gene will not be expressed. So the cell is saying, I do not need this gene to be expressed. I will close it off. And that will be one of the marks that will mark that gene for not being read. The other mark that will decide on whether that gene is read or not is this little green combination of fellas, okay? So what this is actually 8 different proteins called histones. And you have 2 copies of histone H. Well, we won't go into detail, but anyway, you have 8 histones and 146 base pairs of DNA wrapped around them. So what has to happen for a gene to be read is that you have to have assembly of a large number of different modules read that DNA. And you can already see that if this is very tightly positioned together, it's going to be very difficult to read it. So then decision of the cell is to modify these tails of these little fellas as well with certain other chemical modifications which carry also, in addition to DNA methylation, that it's a silencing signal. This could be a number of different outcomes. Sometimes they could say this gene should be switched on and read, and sometimes it could be these should be switched off and not read. So probably the easiest way to view this is to understand this chromatin structure and epigenetic hallmarks by looking at the video. So this is an actual methyl group here, and it sits right on the DNA. You then have the nucleosome, so these are the histone. This is the 8 different little proteins that you saw earlier in green. These are their names for anybody who's interested in detail. Probably not necessary for you to know what this is. And all of these can be modified with these further chemical modifications. So that all in all, that gives a certain signature or a code to that gene. And if we have something known as euchromatin, this is very loose, okay, and lots of signals happen at this level to make this very easy to read and actually create a protein out of this gene. So you can see this is very loosely wound. This fella is actually reading it. It's an RNA polymerase. But if we don't want it to be read, then something really quite different happens. That DNA methylation that I showed you earlier attracts a number of different proteins in the cell to then make this very highly condensed and impossible to read. So you can see there's lots of different proteins arriving. They recognize that methylated DNA. They bind to it and make it very compacted so that actually you can't then read it at all. And between the previous way and this way of regulating genes, this is how cell decides what to do. Oh, sorry. Right, why isn't this going forward? Okay, so what you have now seen is that every person or every animal or any living organism will have a genome that's the same in all the cells, but you're going to have these epigenetic modifications. So these are modifications that sit on top of the genome. And I've just shown you there's DNA methylation, there's modifications, those green little fellas. And then where the nature-nurture thing comes into play massively is the fact that environmental factors such as what you eat, how much you exercise, do you move at all, what's your age? You have already seen that through age, your epigenome must change because how your cells are interpreting your DNA becomes totally different. You no longer look like a child. You're no longer of the same body. And then also ingestion of various toxins through food or smoking or what have you. All of these things affect your epigenome throughout your life. So therefore, the sequence that you are born with has these modifications, but they will change throughout life through environmental factors. So what I'm going to spend the next maybe 10 minutes telling you is how these epigenetic changes change through age in very simple ways, through effects of diet, ingestion of environmental toxins, and stress. So first of all, let's look at age. You all know from your own growing up that age causes multitude of changes, whether you're male or female, it really doesn't matter. That kind of clock, if you like, can actually be read epigenetically. So there are DNA methylations that you can find inside the cells of any person that will tell, without them knowing who you are or what you look like, they will be able to tell by measuring these DNA modifications your age to within 1.5 years, okay? And this is because DNA modification changes at this set of clock genes will change with age, okay? And while this is true for people, it's also true for animals. So what I'm showing you up here, I don't know if you can see the little laser, This is all of the species combined. So all of the species follow the same sort of trajectory. You can tell what age they are simply by looking at DNA methylation in some of their cells. So this tells you that the age drives epigenetic modifications which are easily detectable in all of the species. It also then gives hope that perhaps some of these modifications can be reversed, and therefore age-associated diseases might be able to be kind of reversed. That's the hope anyway. So then diet. Now this is a really, really interesting example, okay? It's one of the things that really brought epigenetics into the forefront because it was everywhere. This story was absolutely everywhere. It came out in 2006, and he was a Canadian researcher who looked at the mouse genome, okay, and decided to actually try and understand how one of the genes within the genome works and how it's affected by diet. So the gene is called a agouti gene, and what it does, it codes or it makes something called a agouti signaling protein. Now this protein, when it's expressed at a high level, will determine the hair color, okay, in animals. So this little fella has a lot of this gene expressed, and that's why the hair is kind of golden color. But this gene does something else as well. It also regulates obesity, metabolism, and susceptibility to disease. So if this gene is expressed, you get a very obese mouse. Now, what they noticed is there are several DNA methylation sites at the start of a agouti gene. So they thought, right, maybe if we populate these with a load of DNA methylation, maybe we can switch this gene off. And one of the ways to do that is through actually supplying in diet a lot of methyl donors. So a lot of these methyl groups that need to be attached here to the DNA, you need to ingest them through your diet. This is why diet is really important. So what they did, they fed pregnant mice with this diet high in methyl donors. And what they then found is that this gene in the offspring became methylated. And what that produced is a mouse that didn't have yellow color, wasn't obese, and it wasn't susceptible to disease. If you were to sequence the genome of these two mice, they are genetically identical, but their phenotype is totally different due to maternal diet during pregnancy. So this is pretty similar in any species. We just use mice in order to understand it. It's easier to do because you need to have relevant controls and find out what's going on. But this is why it's really important to have healthy diet during pregnancy. So talking of ingestion and what we eat and how we eat it, we then come on to environmental toxins. Now these days, most of the things that we eat come in some sort of packaging that is probably in tins. It could be held in some sort of plastic. These are in fact baby bottles. Or we all take our lunches in various plastic containers. And in fact, what all of those plastics have is a chemical called bisphenol A. And that can be ingested in part, just from kind of falling off these particles. You can inhale it. You can assimilate it into your body through skin or eye contact, and it could be transferred onto your fetus if you are pregnant. So that then affects all of these different organs. Certainly, these are the organs where research has found that bisphenol A has effects. And the effect is that this really interferes with DNA methylation. So where you should have DNA methylation, you might not. And therefore, your body starts to make mistakes. Okay? And when body starts to make mistakes, either the cell types are no longer what they should be, and this is how sometimes cancer can develop. So diet is really quite critical, but also exposure to environmental toxins is just as important. So now we come on to stress. How do we handle stress? Is this something that could be transferred on? And one of the best ways, or one of the most recent events where stress was kind of happening for just a short period of time, 4 days, but it kind of went out to be a bit longer, is an ice storm that in 1998, in January 98, that happened over Quebec, around Montreal and the cities here. And it was the most disastrous ice storm ever recorded in Canada. In fact, it's still the worst natural disaster in Canadian history. And what happened is that because it's really, really cold in the winters in Quebec, you get this ice rain, okay? And what that does, it actually kind of builds up on all the trees, and anything that it falls on, it builds up on. So 15,000 trees fell under the weight of ice, because a single tree can accumulate 45 tons of ice during these ice storms. So if an ice storm lasts half a day, that might not be so bad, because ice will melt before the tree breaks. But this particular ice storm lasted 4 days. So there was ice accumulating on the trees, on everything, for 4 days. And it basically just crushed forests, cars, all sorts. It also crushed power lines. So if you're interested in this, please go and watch a video. It's really, you can find it all on over YouTube. You can actually see these power lines collapsing like they're made of matchsticks. It's incredible. But it's under the weight of ice, and they just crumbled. So because the power lines crumbled, they left 4 million people in the dark and without heating. So clearly, amongst the 4 million people, there were a lot of pregnant women. So these pregnant women at that time experienced actual stress. This wasn't subjective stress, this is objective stress, because they were left in the dark, but more importantly, at temperatures of really, really cold. So a lot of people died, didn't make it at all. So then what the scientists did after 1998, they realized, wow, we have a cohort of kids that we could actually see, if you are developing in a mother that's been stressed like this, what does that do to you? So they started a project called Ice Storm, and it's a human model of prenatal stress. So they looked at thousands of children that were in the womb while the massive ice storm occurred, and then they followed these kids and took urine samples and blood samples, and they found that 957 different genes were altered in these kids. And as a consequence, these kids had much more obesity, they had more autism, they had more of different kinds of diseases that were not favorable, because presumably they kind of had this stress in the background. And if you just look at the urine, so this is, these are the male offspring, so these are the sons of the mothers that were pregnant at the time. These were the controls. You can see that metabolites that you can detect in the urine are totally different between the babies that were born to the mothers that were subject to this ice storm to those that were not. So this is presumably due to epigenetic changes that are manifested themselves through alteration of just epigenome during this. So can epigenetics be changed once the animal is born? What I've told you so far is kind of in utero, so you can see how it could be critically important that while you're pregnant, you know, that nothing is happening to the mother, so that that fetus will be affected just by how she carries the pregnancy. But if the fetus is born through a mother that isn't stressed, okay, can you actually change the epigenome? So again, some of the most interesting studies are done in mice. And what you can have is when this is a litter of little mice, okay? So if you have a litter, there are some mums that are very high nurturing. So they are licking the pups all the time. They're kind of handling them, sitting over them, interacting with them all the time. Whereas other mums are just leaving them to one side, maybe feeding them, but not really interested, not really licking them or grooming them or anything like that. And if you then have a look, the offspring of the low nurturing mothers become very stressed and they are anxious adults, okay? Now you could say maybe the reason why that mother is low nurturing mother is because she has some genetic imprint that just makes her that way. And maybe she's passed it on and that's why her offspring are like they are. Now the high nurturing mother has relaxed and happy adults, okay? But the experiment you can do is you can take the litter from the low nurturing mother and cross-foster it onto high nurturing mother, and these pups will become relaxed and happy adults. Which then means the surprising result of that was that the behavior of the pups was not dictated by the genes they inherited from their biological mother, but actually the behavior of the mother who raised them. So the mother's licking and grooming translates into signals in the brain that tell you, I am loved, I'm in a safe environment, my life is going to be good. Whereas the signals that you are receiving from a low nurture is that of stress. So essentially that initial postpartum kind of when you're born period prepares you to understand what your world is going to be like. It might not be the case, but that's how the animals read it and understand it. So similar studies were done in monkeys where they were either left with their mother or separated from a mother onto a surrogate, but the surrogate was only feeding them, wasn't cuddling them. And then if you took the blood, only 14 days later, you could already see differences in their DNA methylation. So some of the genes were switched on in these mice. Sorry, some of the genes were closed off here because they're highly methylated, but they became expressed in these poor little monkeys. And vice versa. Some of the genes that are usually on became switched off. So clearly the epigenome is very plastic. It can change very readily, even over a period of 14 days. And the reason for these high nurturing behaviors to be so important is because it leads to brain development where the brain is producing greater serotonin levels. And I'm sure that everybody's read why serotonin is important to keep you kind of happy and depression-free. Now, this is a really interesting study that maybe younger students might really enjoy. I know my son will enjoy this in the background. I'm hoping he's not up to this sort of thing when he goes out. But one of the studies that were done is to expose rats to a 10-day cocaine party, okay? So they took two groups of rats and they let them binge on cocaine for 10 days, okay? Then they withdrew the cocaine. And what they did, they left them for a month for the withdrawal to happen. And then one group was re-exposed to cocaine where they went, so initially the cocaine came through here. So they liked it, they ate it. As soon as they went back into the cage, they were pressing the lever, okay, which dispenses cocaine, right? Because they were just having this as a pleasurable thing, great. And they kept pressing and pressing the lever until they actually overdosed because they developed dependency and liking for this experience. At the same time, a different group was also left for a month, but they were given a DNA demethylation treatment because what they found in this group is that during that month, the brain was rewired to start really, really craving cocaine. And so as soon as they could, they started pressing the lever and eating more and more and more cocaine. But once they had this DNA demethylation treatment, the rats were no longer interested. They weren't addicted anymore. So you can actually reprogram your brain both through overuse of something, but also through using some of these epigenetic treatments to erase the behaviors that have happened in your own lifetime. So what I've shown you so far is a result of pregnancy or early life experience, but there's kind of several things here. Can you inherit epigenetic signature across generations? For example, does it matter what the sperm and egg go in with? And if any of you are biologists here, you will know there's a dogma that says that once a fertilization happens, that the slate or your DNA is completely erased of all epigenetics marks, okay? So you're starting from scratch. This is not true, okay? This is really, really not true, and I'll show you why. So we're going to talk a little bit about epigenetic inheritance and how what your grandmother or grandfather did really makes a difference to you and your lifestyle. But I will also show you that you're not necessarily stuck with that for the duration of your life. So one of the experiments was done in mice, and this is very much like Pavlovian dogs. If you have ever read works of Pavlov, he was exposing dogs to certain stresses in order to understand how that stress was affecting their personalities and stuff. And often there would be this association of two different triggers so that if only one trigger was present, it was triggering the other one automatically in the brain. So this is that sort of study. And what they did is they exposed mice to smell of cherry blossom, but while they were smelling the cherry blossom, they also gave them an electric shock. Okay, I know it sounds nasty, but you know, it's quite, these things have to be done at times. So this is a classical conditioning experiment, okay? You start to associate the smell of cherry blossom with fear because you know what's coming, okay? You know you're going to get electrocuted a bit, zapped a bit, the minute you smell it. So if we look at, so the brain waves can be recorded during electric shock. And what you see is that the brain waves really change, okay? So this is high stress situation. So this animal has got this learned behavior. I smell cherry blossom, I know I'm going to get electrocuted. And then once they were conditioned, they were then exposed just to cherry blossom, okay? No electrocution at all. And the brain waves were the same, okay? So just exposure, just smelling cherry blossom gave them high stress response because they were already conditioned to associate fear with that smell. Now, if you took one of these animals, and so this is a conditioned animal that has been electrocuted himself, but is now responding just to cherry blossom smell. And if you bred him against a female that was just completely normal, she didn't have any stress responses, she had normal pregnancy, she was nurturing the young and everything else. So these babies that came from this dad had no exposure to anything at all. If you then expose them to cherry blossom smell, their brainwaves were the same. They had the same stress and fear response at smelling cherry blossom, which then means that something has passed from the dad onto these offspring. And the only thing that male provides is sperm. So there's no in utero, there's no pregnancy, there's no nothing. So it's just what's in the sperm is actually informing the animal, be careful, when you smell the cherry blossom, danger, okay? So just like the mother's behavior and her experiences are passed onto the pup, Same works for a man. So it's really important what you do in your young lifestyles before you go and having kids. So now we're coming on to some of the work that I've done, and I'm going to make it massively simple, okay, because it was done over many, many years with a lot of complexity, but I'll try and bring it to you in a highly kind of reduced form. So basically, what I was working on is liver fibrosis. Most of you will know if you drink loads and you keep on drinking, you're going to get scarring of the liver. So a lot of people will eventually kind of have this. They might have some tests done, and they're going to be going, oh my God, you know, I'm in a terrible state. I'm going to have to stop drinking, or I'm going to have to stop eating, or I'm going to have to lose weight. There's all sorts of reasons why you might develop scarring in the liver. But we kind of wanted to have a model where we could understand whether any of those events that happen during kind of liver fibrosis or scarring of the liver could be mediated to the offspring in much the same way as what I've explained to you for cherry blossom and stresses, okay? And what we did is we said, okay, if we have two different groups of rats, okay, so we're starting out with same thing. If we then make these ones in red experience an episode of having liver fibrosis because they were, say, binge drinking or something like that, okay, equivalent in a person. And if we then breed them to produce sons, again, so we're just going through male lineage here, okay? We're just testing sons. So if we then split the sons into two groups, okay, so this grandfather, we'll call him, well, father in this sense, his sons are split into two groups, and these guys that have had an episode of liver fibrosis but recovered partially, they also had sons. And then what we did is repeated the same thing. So if you now injure these and injure these, if everybody's the same, what you should get is same amount of fibrosis, okay? But you don't. So what happens is that despite these guys being injured, okay, they seem to have remarkably normal liver, as though the father has somehow informed them how to respond to liver injury, okay? Now, these ones, to all intents and purposes, controls. So no injury, but when you injure them, all of a sudden you have really profound liver disease. So any combination of father or grandfather having episode of kind of damage seemed to invoke liver being quite normal. So that made us think, well, how does that work? How can we find out what's actually happening? And clearly there was something in the sperm, again, that was being passed down. But how could we find out? Because this animal is injured himself, so maybe that injury goes to the sperm anyway. This animal is injured as well. So the only animal that isn't injured is this one, but it has ancestral lineage where there was damage. So we then bred these animals as well, produced their offspring, and then we injured everybody. And what we found is that, again, you had all of these were protected against developing fibrosis, but this top one is the only one that got injured, again. Now, what I'm kind of drawing your attention to this particular group here, these rats were never injured, but they gave rise to a progeny that was also protected, okay? So they must have had some sort of memory from the granddad that gave them that protection. And if we then try to understand what that protection is by comparing sperm from these guys and these guys, we can see that DNA methylation, again, is very different in the sperm if you have no ancestral fibrosis versus if you have fibrosis in the grandfather. So what your grandfather did, as well as what your father did, really matters, okay? Now, is there anything of that in people, all right? Do we ever see any of these events in people? Because obviously, laboratory experiments are done in highly controlled ways. You know, does this really have any relevance to what people do? Well, we thought yes. And the reason is that if you take 100 people, could be students, could be older people, it doesn't really matter. And if you get them to binge drink, like really do some, and everybody's drinking exactly the same amount, there's going to be only very few, actually, that develop fibrosis, and of those, even fewer that will develop end-stage cirrhosis. And that number is around 15 to 20% of people will go on to develop cirrhosis. So it seems like the genomes are pretty similar across all of these groups, but the epigenome might not be. So how do we study that? So the best way to study that is actually to find a group of people that have been having whatever behavior, okay, for some time but remain stable. And then another group that's comparable who have rapidly progressed to have liver cirrhosis and were either transplanted, were given new liver, or they died. So that's exactly what we did. We looked retrospectively at people who, right at the onset, so this was back in the 90s, they had liver biopsy taken for a variety of reasons. So we had a picture in time of what their liver was like, okay, 30 years ago. And 30 years later, we know that these people were still the same because they had another liver biopsy. And we know also that these are either dead or transplanted. So at the starting point, none of these people had anything you could see in the liver that was different between the two groups. But when we looked at their DNA methylation in the organ, there was already a signature over 487 different genes that had more methylation than they should or less methylation than they should. And that seemed to predispose them to either developing cirrhosis, dying or being transplanted, or staying normal for the following 30 years. So I hope I've managed to convince you through animals and some of my own work in people that really it's quite critical what your parents and grandparents do. They pass down to you much more than their genes, but there are epigenetic drugs, there are things you can do in your own lifetime that can change these things. And mums are there not only to pass the genome, sometimes epigenome, but quite critically, pregnancy and in utero experiences. So for lads, choose the woman you're having the kids with very carefully, and vice versa. So again, we come to that, lifelong question, nature versus nurture. I would say they're equally balanced, okay? You could overcome really great nature by having terrible nurture, eating really badly, doing bad things, smoking, drinking, behaviors that are not conducive to having healthy life. So I hope I've managed to convince you that our DNA provides a script and that epigenetics allows for a dynamic narrative in our life's movie. But it offers you not only a degree of freedom, but also responsibility over your biological destiny. Thank you very much for your attention.